Hello everyone, here we are discuss about self-incompatibility. Self-incompatibility was first reported by Carl Reuter and it is defined as inability of plant with functional pollen to set seeds when they are self-pollinated. It prevents self-fertilization and encourage outcrossing and allogamy. Due to outcrossing and allogamy, it reduces homozygosity and promote heterozygosity. It may occur at any stage through pollination to fertilization. Self-incompatibility has been reported in 66 families of plant. Now, what is the genetics of self-incompatibility? There is a most popular hypothesis called opposition as allele, which was proposed by East and Mengeldorf in 1925. According to this hypothesis, self-incompatibility due to S gene. S gene has more than 2 alleles in a population. These alleles are designated as S1, S2, S3 and they are present in different genotypic combinations of plants in population like S1, S2, S1, S3, S2, S3, etc. Morphologically, self-incompatibility is of two types: heteromorphic self-incompatibility and homomorphic self-incompatibility. Heteromorphic self-incompatibility is further of two types: distyly and tristyly. Homomorphic self-incompatibility is also further divided into two types: sporophytic self-incompatibility and gametophytic self-incompatibility. Now, heteromorphic self-incompatibility. As the name indicates, heteromorphic, which means it is due to difference in the morphological feature of the flower. So, this is type of self-incompatibility which is due to difference in the morphological features of the flower. It is of two types: distyly and tristyly. First, distyly. Distyly, as the name indicates, stamen and styles are of two types. As shown in the diagram, there are two types of flowers: pin type and thumb type. Pin type flowers having long style and short stamen, and thumb type flowers having short style and long stamen. When thumb flower is crosses with the thumb flower, they are incompatible to each other. But when thumb and pin are crosses with each other, then they are compatible for each other. And most important thing is that it is determined by the single gene with two alleles. Example in primula. Second is tristyly. Also here the name indicate that style and stamen have three different sizes. They may be small, medium and large as shown in the diagram. It is also controlled by two genes S and M, each with two alleles. Example in lithium. Now, homomorphic self-incompatibility. Again, name indicate homomorphic means flowers having similar morphology. So it requires proper breeding test for their recognition. They are controlled by single gene as having multiple alleles. And it is also further divided into two types: gametophytic self-incompatibility and sporophytic self-incompatibility. Now, gametophytic self-incompatibility is different from sporophytic self-incompatibility. Gametophytic self-incompatibility is determined by the genotype of the male gametophyte, and sporophytic self-incompatibility is determined by the genotype of the sporophytic plant. Most important thing, gametophytic self-incompatibility, in this pollen rejection occur in the style of the gynoecium, but in sporophytic self-incompatibility, pollen rejection occur in the stigma. In In gametophytic self-incompatibility, pollen grains are unable to germinate on stigma with similar allele as that of pollen. Example in Liliaceae, Poaceae and Solanaceae. As shown in the diagram, the pistil having S1 S2 alleles and the pollen which having S1 and S2 alleles are not able to germinate on the pistil which having S1 S2 alleles. In the middle diagram, we can see that the pistil having S1 and S3 allele and the pollen which having S1 allele is not able to germinate, but the pollen which having S2 allele, which are totally different from the alleles which are present in the pistil, so it is able to germinate. In sporophytic self-incompatibility, pollen does not germinate on the stigma of a flower that contain either of the alleles that are present in the sporophyte parent which produce the pollen. Example in Brassicaceae and Asteraceae. Sporophytic system, as we show in the diagram, seen in the diagram, the pistil which having S1 and S2 allele and the pollen which having S1 S2 allele is not able to germinate. In middle, the pistil having S1 and S3 allele and the pollen which having S1 allele not able to germinate, but the pollen having S2 allele is also not able to germinate. In the last diagram, we can see that the pistil having S3 and S4 allele and the pollen which having S1 allele, which is totally different from the pistil, is able to germinate, and the pollen which having S2 allele is totally different from the alleles which are present in the pistil, so able to germinate. Now, physiology and biochemistry of incompatibility. There are two reactions occur. First is recognition reaction and rejection reaction. In recognition reaction, here pistil recognize a pollen, and after recognition it accept or reject the pollen. So recognition reaction is occur at the molecular level. By this, pistil recognize whether a particular pollen grain is to be accepted or rejected. In rejection reaction is occur on the stigma or style. In stigma, inhibition of pollen germination or prevention of the entry of the pollen tube into the stigma occur. In style, inhibition of pollen tube growth or leading to the bursting of tube in the style occur. The pollen wall and the protein content in the pollen stigma interaction play a very important role. Stigma surface inhibition. In sporophytic self-incompatible system and some gametophytic system, the recognition and rejection reaction occur at stigma. So, as shown in the diagram, there are pellicle layer, cuticular layer and callosic plug are help for stigma surface inhibition. So, above the cuticle layer, pellicle is present, and then cuticle is present. They are present discontinuously, and then pectocellulosic wall is present. So, pellicle layer, it is hydrated layer. Here, pollen grain come, and the pellicle layer act as a receptor for the proteins which are present on the surface of the pollen grains. Here, interaction of the protein which are present on the surface of the pollen grain occur by the receptors with the receptors which are present on the pellicle layer, and they recognize that the particular pollen grain is compatible or non-incompatible. Cuticular layer. Compatible pollen grain able to penetrate the cuticle layer. So, to penetrate in the cuticular layer, Cutinase enzyme is very much necessary. So only if there is compatible pollen grains are present, then cutinase enzyme, which is necessary to erode the cuticle, is activated, and by this the pollen tube able to penetrate the cuticular layer. Callosic plug. If there is incompatible pollen grains are present, then there is a formation of a callosic plug between the plasma membrane and pectocellulosic layer, which sees the pollen tube formation as shown in the diagram. Now, styler inhibition. In gametophytic self-incompatible system, recognition and rejection reaction occur in the style. There are three processes: peroxidase activity, RNase activity, and bursting of pollen tube occur. Peroxidase activity. There is much higher activity of peroxidase than in style with incompatible pollen tube. It was seen in Nicotiana alata. Second is RNA activity. In incompatible pollination, RNA is present in the style. It penetrates into the pollen tube and degenerates the RNA in the cytoplasm of the pollen tube and inhibit the pollen tube growth. Bursting of the pollen tube. In incompatible style, the tube initially shows normal growth, but after 2-3 of the length of the style, inner wall gradually thin down and eventually disappear. Simultaneously, small particles in the tube cytoplasm appear and the tube get filled with these particles. The outer wall become thickened and give swollen appearance. And at this stage, pollen tube get bursted. So these are the styler inhibition methods occur in the style. So methods to overcome incompatibility. There are different methods: mixed pollination, in vitro pollination, heat treatment, irradiation, and increase in CO2 concentration. Mixed pollination. It is like a camouflage. In this, stigma is pollinated with a mixture of compatible and incompatible pollen grains. So compatible pollen grains provide recognition protein which permit incompatible pollen to germinate. In vitro pollination, placing the pollen grains in direct contact with ovule. Heat treatment. By giving the high temperature varying from 30 to 60 degrees leads to breakdown of the self incompatibility. Irradiation of style with X-ray immediately before selfing, resulting in the breakdown of the self incompatibility. An increase in CO2 concentration is reported to overcome self incompatibility in the sporophytic system. Importance of self incompatibility. Self incompatibility effectively prevents self pollination. As a result, it has profound effect on plant breeding approaches and objectives. Self incompatibility used to make hybrid seed production. Self-incompatibility provide hybrid seeds without emasculation and without resorting to genetic and cytoplasmic male sterility. Self-incompatibility system increase the probability of new gene combinations. Limitations of self-incompatibility. It is very difficult to produce homozygous inbred lines in a self-incompatible species. Second thing, bud pollination has to be maintained. Third is self-incompatibility is affected by the environmental factors such as temperature and humidity. And it is very tedious and a costly process. Thank you.